Plant anatomy. Plant anatomy means study of internal structure of the plant body. Then how to study internal structure? They are not outside. They are internal part. So to ensure that we make a section cutting, we do section cutting. Through the section cutting, we can observe the internal parts with help of the microscope. Now, recently, most of the advanced microscopes are available to study in detail about the tissues, about the histology, about the cell. The mainly in anatomy, we we study detail about the cell structure, tissue, and the total histology. So, what is cell? First of all, the cell is the basic unit of the organism or body part, and it is a structural and functional unit. This group of cells together form as a tissues. The study of tissues and tissue organization, these are all called as histology. So these are all we are going to learn in detail in plant anatomy, right? So the plant anatomy, study of internal structure, number one. We study or examine internal parts after the section cutting of the part, either this transfer section or vertical section, whatever it may be, any manner of section cutting, we observe the tissues or internal parts through the microscope, right? So what you examine through the microscope, mainly the type of cells, tissues, and organization. These are all. So already you know the cell definition and tissue definition and histology. Let's see first of all how the formation of tissues. Mainly in anatomy, we focused on tissue, right? Study of tissues, the tissue that is made up of group of cells. And if you take, for example, let me take sketch. Right. So if you take the first of all, before formation tissue, after fertilization, male and female gamete fusion, here formation as a zygote, single cell. The zygote is the single cell. Of this single celled zygote, later further divided and subdivided by mitotic divisions and form as a group of cells. This is group of cells, right? So group of cell, not a single cell. It is made up of number of cells. First of all, the single cell part is the only zygote. This is the single cell, which is formed after the fertilization of male and female gamete. Then, then this single cell zygote undergoes to mitotic division. Later, they forms as the group of cells like this. In this, these cells called as embryonic cells. These cells called as embryonic cell. This embryonic cell, the further divided in division, one embryonic cell, E for embryonic cell, divide into two cells. One is meristematic cell, another one is permanent cell, like this. So like this embryonic cell, the permanent group of cells together form as a permanent tissue. And here the meristematic cells, group of meristematic cells form as a meristematic tissue, right? So anyway, after formation, the permanent and meristematic tissue from the embryonic cell, the permanent tissue cells cannot divide further. But here the only meristematic tissues, here the only the meristematic tissue cells divide, redivide, and forms again meristematic tissue cells and again permanent cell. Meristematic cell and permanent cell. This meristematic again divide and again form meristematic permanent. Like this, the number of permanent cells altogether formed. That is called permanent tissue, right? So, but the meristematic tissue, they have the capability of division. But permanent tissue does not have capability of division. But whenever it necessary, they can regain ability of division. The ability of division in permanent tissues also, but mainly the division capability only present in meristematic tissue, but not in permanent tissue, right? Let's discuss the meristematic tissue. Before that, here the just small comparison. Already told you the small comparison between meristematic tissue and permanent tissue. Meristematic tissue able to divide, but permanent tissue temporarily not, but not permanent, temporarily not, right? And again, the major part of the plant body made up of permanent tissue because they are deposited in the plant body. So, and this permanent tissue again divided into simple tissue and complex tissue. Simple and complex tissue, and also apart from the complex, the some plants have the special tissue also present. Special tissue also present right down here. Apart from the simple, complex, and special tissues also present, but it is not given in our syllabus. But remember it, the special tissues also present, right? So permanent, the simple, in simple tissue, parenchyma cells, collenchyma cells, sclerenchyma cells. The complex xylem and phloem. So first of all, let me complete in detail about the meristematic tissues. Meristematic tissue. After meristematic tissue, because it is the first form tissue, after meristematic tissue, now let's we enter into the permanent tissue. In permanent tissue, the first we will going to complete simple permanent tissue, complex permanent tissue, later specialized tissue. Okay. The difference between simple and complex permanent tissue. Simple permanent tissue, they are the group of cells are similar in structurally and functionally. But in complex permanent tissue, this in name itself given complex. They are in complex. They are not alike in a structural. Of course, function same. Altogether perform a same function, but they are structurally different. So that is called as a complex tissue. And specialized, they are tend to be the special. They are present in some of the family but not all. One of the best example, the latex production. The production of latex, the milky substances exuded out, it is done by specialized tissue. It is not present in all plants. They are the only the limited plants the specialized tissues are present. Sorry. Now let's see first meristematic tissue, after that permanent, right? The first meristematic tissue, this is very important for essay question. Meristematic type of meristematic tissue. Type of meristematic tissue. So if in examination they ask type of meristematic tissue, simply we write what is meristematic tissue definition. After that, types. Right? The definition meristematic tissue already told you they are the small cells, group of cells, they are in small and they have the capable of division without intercellular space. So without intercellular space means there is no gap between the tissue of the cells. So that is called intercellular space. And nucleus also present inside the tissue. Mainly it have the active division because of the consisting of mitochondria, number of mitochondria. And vacuoles very less and a small vacuole present or maybe absent in meristematic tissue. So this is the small introduction about the meristematic tissue and definition. Let's see the types. Actually, the types of the meristematic tissue based on position, based on position, based on position and based on division. Not given here, but remember one is position, second one is division. First, let me complete about the position. And also based on function also. I will explain later function. Based on the function. But it is not important. Yes. So first of all, if you see based on function, sorry, based on position, they are clearly divided into, if you take, let me erase these are all. Position, name itself given where they are. Maybe they are apical, maybe they are intercalary, maybe they are lateral. Right? Just wait. Right here. Right. So now the classification of meristem, meristem definition clear. Now the classification of meristem. Uh, right. Based on origin, already told you based on origin or position. Number one, origin, and another one is position, and second one is plane of division. Plane of division. First, origin, promeristem, primary meristem, secondary meristem. Promeristem in embryonic cell. From embryonic cell, after division, the primary meristem formed. Embryonic cell considered as a promeristem, which is divided and formed as a primary meristem. Already told you, after formation of primary meristem, uh, after formation primary meristem, just wait. Right. After formation primary meristem, this primary meristem again divided into two cells, again form a meristem, primary meristem, and another form as a permanent cells. But I already told you the characteristic nature of the meristematic tissue have the capable of division, but characteristic of the permanent tissue not have the capable of division. But whenever it necessary, they can have the regain the capable of division. That's why sometimes what happens, this permanent tissue whenever necessary, they can they can metamorphose into meristemic tissue. So this is formed second time. That's why this is called as a secondary meristematic tissue. So this is called as a secondary meristematic tissue. The first formed primary meristematic. Promeristem in embryo. From the promeristem, primary meristem. After primary meristem, sometimes the permanent tissue also act as a meristematic tissue, that is called as a secondary meristem. The function of secondary meristem and formation of secondary meristem, I will explain later in secondary growth. Right? Now position. Now position. Based on position, apical meristem, intercalary meristem, intercalary, intercalary meristem, and and lateral meristem. Excuse me. Sorry. Right. First apical. Name itself given apical. So the apical meristem, which is present at the apical side of the stem, apical side. This is called apical meristem. The function of apical meristem, it shows it is involved in the growth of the plant body, linear growth, like this, involved in linear growth of the plant body. So plants grow the entire life due to the activity of this apical meristem tissues. Next, intercalary meristem. This intercalary meristems deposit at the nodes and internodes region. Here, this is intercalary meristem at the node region. Intercalary meristem mainly present in the grasses family. For example, this is the stem. This is the stem, node, internode. This is also node between the place internode, and this is the apical part. So if the meristem present in apical position, that is the called as apical meristem, and it is involved to the linear growth, right? And this is the node region. If the node region meristematic tissue is also present, and it is also involved in for the growth, involved for the growth. So if the present at the node region, that is called intercalary. So this is intercalary meristem, and last one, lateral meristem. Lateral means side. This is lateral side. This side, partial side. So this is called lateral meristem. Actually, this lateral meristem present inside the vascular bundle. If you take section cutting, for example, this is the section cutting. So these are the vascular bundle, for example, vascular bundle. So if you take one vascular bundle, here clearly one vascular bundle, meristematic tissue, inside xylem, outside phloem. So this meristem called as lateral meristem. This meristem called as lateral meristem, and this meristem functions also growth, but not linear growth. It is involved in for radial growth, like this growth, radial growth. Apart from the linear growth, radial growth also required to sustain the plant body, to carry the plant branches, to weight the able to weight the branches. It need to be elaborate by radial growth. So which is done by lateral meristem through the secondary growth. I will explain later secondary growth. Right? This is only position. And next one, plane of division. Next one, plane of division. Plane of division, the meristematic tissues again divided into three types: mass meristem, plate meristem, rib meristem. Simple. Mass, plate, rib meristem. First of all, rib meristem. First, let me complete first one, rib meristem. In rib meristem, this is the cell. This is the cell. The cell divide like this, periplic anticlinal division. This division called as anticlinal. This anticlinal division due to the anticlinal division, the layers are formed. For example, here the three cells are there. The three cells the same manner divided, which means inside three formed. Again the same division, again three formed. Like this, the cell tissue layers are formed due to the rib meristem. So by this, in plant body, mainly formation of cortex cells. Cortex cell made up of more than one layer of tissues. Cortex. If you cortex and ground tissues, that means vascular bundle in steel part also by meristematic tissues. Some of the steel part, right? For example, if you take internal structure of the plant body, this epidermis, rest of the part called as cortex, which is made up of cell layers like this. For example, this is cortex. After cortex, this is the steel, and after cortex, steel part is present. So steel also have the layers. Central medulla, central of the medulla. So this is all done by rib meristem. Next one, plate meristem. It is shows the periclinal division. Periclinal division means the same cell. Let me try here. Same cell that divide like this. That is periclinal division. This is anticlinal division. What happened if division like this periclinally? Periclinal layer surface improved. Not that means the cell surface improved. Only cell surface improved, but not layers. But not layers. Like that, here the mainly formation as a epidermal cells. Epidermal cell single layer like this. It is like this epidermal cell. So this epidermis growth done by plate meristem division. Division of plate meristem. And cortex cells inner parts mainly made up of rib meristem. And mass meristem, there is no proper way. It can irregularly division. Mainly it is formed in medulla, center of the part. Irregular division. Irregular division is the center of the part. For example, this is the center. This is the center. In diagram, this is the center. So in the center, irregular cells are formed. So this is done by mass meristem. And the rest of the by done by rib meristem. And final, this epidermis, this final layer, single layer epidermis done by plate meristem. So this is the based on division. Right? Is it clear? So definition. First definition. The group of cells which are small in size without intercellular space have the capable of division. Have the capable of division. And it is classified into mainly based on origin and based on position and plan of division. Plan of division. Origin, they are promeristem, primary, secondary meristem. And based on position, they are apical meristem, intercalary, and lateral. Plan of division, rib meristem, plate meristem, and mass meristem. Right. This is about the meristem types. This is meristem types. Now let's discuss about the theories, apical cell theories. This is also very, very important. Apical cell theory also important. Let's see another slide. Just wait. Apical shoot apex theories and root apex theories. First, let me complete about shoot apex theories. That may how the shoots are developed according to some scientists. After that, root apex theories. Root apex theories and let's see the shoot apex theories here first of all. Uh, right. Shoot apex theory. Shoot apex theory. Let me take pen. Apical cell theory, estrogen theory, tunica corpus theory. These three important to explain about shoot formation, shoot apex theories. First of all, apical cell theory. The apical cell theory proposed by Hofmeister, and this theory supported by Nageli. In this, only single cell is the structural and functional unit. That means this is the shoot apex. In shoot apex, only one cell is present, active. This one cell divided and redivided form the tissues, and and finally it forms the parts of the body. So this is the one type of growth which is done by only single cell. Next theory, estrogen theory. Estrogen theory proposed by Hornstein, supported by Strasburger. It is not a estrogen. Estrogen means not a single cell, the group of cells. The same here, I will show the diagrammatically later. Same apex without a single cell, the group of cells are there. The group of cells comprise three distinct zones. Let me change color. Uh, One, right. One, two, three. These three group of cells are there. The one is dermatogen, as it given here, the dermatogen first layer, dermatogen, this one. Second, peribulum, this one. And third one, plerum, right. Dermatogen, it is the outermost layer and it forms the epidermis by the anticlinal division. Sorry, by the periclinal division. By plate meristem. By plate meristem like this, this is the cell periclinal division. Due to periclinal division, cell surface layer increased by formation of epidermis. Next one, peribulum. Peribulum. It is the middle layer. This is peribulum, and it gives mainly cortex. Already told you by anticlinal division like this. This is the cell. Plerome. Last third one. Plerome. This one. Plerome is the innermost layer. Plerome is innermost. Plerome. Plerome is innermost. Right. Sorry. Plerome is innermost layer. It gives the steel part of the plant body, and which is also by either same by rib meristem like this or mass division like this is irregular division. So this is estrogen theory, and most of the scientists accepted theory tunica and corpus theory. Tunica corpus. Tunica corpus theory, which is proposed by Smith in 1924. According to tunica corpus, only two zones are there. The two zones, one is tunica, another one is corpus. Instead, rather the three, here the only two. One, two, that's it. Two zones. This is the tunica, this is the corpus. The tunica is the peripheral zone, outer zone of shoot apex and forms the epidermis. The corpus is the inner zone apex that forms the cortex and stele of the shoot. Both cortex formation and stele formation of the both internal part of the plants. So this is the shoot apex theories. Three important: apical cell theory, estrogen theory, tunica. But the most accepted theory is tunica corpus theory. Let's see the diagrammatically now. Right. If you see shoot apex theories, first A apical cell theory, B estrogen theory, C tunica corpus theory. Actually, in this apical cell, okay, 1 minute. In this apical, of course, leaf primordia formed, but here the only one active cell, the blue color one, this one. Only this one active cell. This one active cell structurally, functionally active and divide, redivide from the tissues, from the apical tissues. So this is the shoot apex theory, shoot apex, and mostly this is applicable to the lower plants like pteridophytes, bryophytes. Mainly the lower plant perform this type of growth. And next one, estrogen theory. The group of tissue, group of cells actually classified into three regions. Outermost dermatogen, middle peribulum, plerome. This all together constitute called as the estrogen. Right? The see you can see this diagram only two regions, outer tunica, inner corpus. That's it. From outer corpus only epidermis formed, the rest of the body parts developed from the corpus region. So this is the diagrammatical explanation about cell theories. Now root theory, of course, gymnosperm cell theory, pteridophyta cell theory also there, but it is not important here in according to our syllabus. These three important. Next one, shoot apex theories. Sorry, root apex theories or shoot apex theory completed. Root apex theories, root apex theory. Let me take pen. Here also same like a shoot apex. Here also same estrogen theory. And next one, mantle core theory. I will explain it. First, let me complete estrogen theory. Same, it is present in shoot apex and root apex, which is proposed by Alstein and supported Strasburger. Estrogen theory is applied to the root apical meristem, speaks the four estrogen meristem here. But in shoot only three meristems, region, but in root four region. Let's see what is the extra region here. Dermatogen common like a shoot apex, peribulum common, shoot apex, plerome common, and also function also same. Dermatogen outermost gives epidermis, peribulum cortex, plerome steel, plerome steel. Fourth extra one, the protective layer, that is the calprogen. It's from the root cap. It formed this one. It formed the root cap. In shoot, there is no cap, but in root, to penetrate into the soil surface, they need one cap, strong cap. That is the it is made up of root cap. So this is about estrogen theory of root apex. Root apex theories. Next one, mantle core, mantle and core, corpus cappe. Let's wait. Carper cappe, mantle core actually right. Mantle core not given. So whatever it may be, Carper cappe. And apart from the mantle and core look like same Carper cappe theory. Now another one, Carper cappe theory, which is proposed by Shuk. Carper one region, cappe one region, root apex, Carper, body, Carper means body, cappe zones form the cap. Simple. This cap, root cap formed. This theory is equivalent to the tunica carpus theory. In shoot, tunica carpus look like tunica carpus here also the Carper cappe theory formed. The two divisions are distinguished by this type. One is T type of division, another and also called Y division. Carper is characterized by inverted T or inverted Y, Kappa by straight die. For example, this is inverted division here, like this inverted division. But here, straight D. Body, the body T, the Carper characterized by inverted. This is the Carper. So actually, right, this one. Kappa zone. This is the body, Carper, and this is root cap formation, Kappa zone. And look like the Carper Kappa and another one mantle core theory also there. Mantle core was only it given mantle and core two regions. One is the mantle and another one is core. Mantle is outer part, core is the center part of the body. And quiescent center concept. It is a very important quiescent concept. The actually this was proposed by Cloves to explain root apical meristem. Root apical meristem activity. This center located between the root cap and differentiating cell of the roots, for example. Root cap, this is the root cap, the entire part is the root cap. This is the cells differentiating region, cortex T, like that, this part, entire part. Now this center one, this is called quiescent center. This is called quiescent center. Actually, this apparently an inactive region of the cells of root meristem is called quiescent center. They are not a active, they are inactive cells. It is the site of hormone synthesis and also ultimate source of meristematic cells of the meristem. But it is also meristem, it gives the meristematic cells. Gives the meristematic cells. So this region called as quiescent center. Right. And any another important in your book, whether given or not, about quiescent center. Let's see. Let me see. Suitabex theories. Okay. Right. That's right. Then what is Suitabex theories? Okay. Right. But not given. Right. Quiescent center. Right. Quiescent center is the short answer. Important. Right. Next one. Now the tissues. Permanent tissue. What is permanent tissue? Already told you permanent. It is a group of tissue, and they are not have the capable of division. Right. Not have the capable of division, and they are again classified into, as I said earlier, into let simple permanent tissue, complex permanent tissue. First, let me complete about the simple permanent tissue. This one, this region, simple permanent tissue. Simple permanent tissue. The definition simple permanent tissue, the tissue itself, group of cells, but their simple means all tissue cells are similar. Structural similar, functional also same. So that is called simple tissue. And in this, parenchyma, collenchyma, sclerenchyma are there. Now first let's discuss about the in simple permanent tissue. First of all, let me complete about parenchyma cells. Right. So this is the parenchyma. Right. Parenchyma cells. Same simple tissue, simple parenchyma definition. The parenchyma cells are the group of cells all are similar structurally, function also same. The common they are the oldest cells and fundamental tissue. It is the parenchyma tissue is the fundamental and basic fundamental tissue, and it is present in all type of plant pots. And it is developed in algae and bryophyta, pteridophyta. That's why it is called one old and simple one. And let's discuss this occurrence. The parenchyma is commonly seen in cortex of the stem and root, medulla region, pericycle, mesophyll cells, mesophyll spongy, palisade, embryo, endosperm, and xylem parenchyma and phloem parenchyma. This is they are the place. And let's see the origin. Origin actually before parenchyma, first of all, the permanent tissue, the origin from the meristematic tissue. Remember that one. And parenchyma, primary body of cortex and pith or mesophyll cells. This is the origin place. If you see the structurally, parenchyma cells usually isodiametric or polygonal or round in different type of shape. And they are usually thin walled layer. We can see diagrammatically thin layer. Here, this is the cell and cell covered by layer like this. This is the cell and covered by thin layer, covered by thin layer, covered by thin layer, and with they are living cells because they have the cytoplasm and all cell organelles, and it have the intercellular space. We can observe here this gap. This is intercellular space. Let me mark it into. This is also intercellular space. This is also intercellular space. So it should have the intercellular space, right? And chemically, it is made up of cellulose. It is this chemical structure of the cell wall of parenchyma, cellulose or hemicellulose. And parenchyma cell shows the considerable variation in their structure. They have thick walls and storage tissue. Maybe based on this, actually different type of parenchyma tissues are there. One is storage tissue, and the that is called storage parenchyma, and another one is chlorenchyma. Chlorenchyma, another one is aerenchyma. Simple storage, chlorine, chlorine, aren, aerenchyma. And lots like that, the different type of cells, and also storage parenchyma, water storage tissue, and water storage parenchyma cells. Also, these are the different type of parenchyma cells. This basic is same like this formation. If it is storage, these tissues this, with filled with food material. That is storage tissue. When compared with other parenchyma, the storage tissue cells made up of thick cell wall. Cell thick cell wall. It is made up of thick cell wall, somewhat thick when compared with other type. So inside the cells, the food material stored. So this is called storage parenchyma. Mainly, you can assume the storage parenchyma tissue where the storage part mainly endodermis, embryo. So the mainly the endodermis, this type of cells are there. And also next one, aerenchyma. The name itself given air. So the cells not well closed in aerenchyma, not well closed. They are the maintain the vast gaps of cell intercellular space. intercellular space. Due to the vast gap, this intercellular space filled with air. This type of parenchyma called as aerenchyma, you can assume this type of aerenchyma mainly present in water hydrophytes, water plants. So this is called parenchyma. And another one is storage parenchyma. Sorry, another one is chlorenchyma. I forgot this chlorenchyma. Chlorophyll. Chlorophyll, for example, this is the chlorenchyma type of tissue. In chlorenchyma type of tissue, let me take green in color. Here, what happened? The pigments are present like this. Chlorophyll pigments are present. If the chlorophyll pigment deposited in the parenchyma cell, that is called as chlorenchyma. It may be spongy or palisade. The function of this storage parenchyma function, storage food material. What is the function of chlorenchyma? As it consisting the chlorenchyma cells, the function in involved in photosynthesis to prepare food material, right? And aerenchyma with filled with air, filled with air, it is mainly present in hydrophytes. Water storage. You can see that mean already name itself given water storage. That means the parenchyma cell storage the water. The best example, aloe vera plant. If and agave plant, if you break the aloe vera, the jelly substances come out. The gel substances, that is entire tissues. The gel is made up of the parenchyma type tissue. We store the water. We store abundant of water. It appears as a gel substances. It is mainly present in desert plant, xerophytic plant. Xerophytic plant. So this is about types of parenchyma. Is it clear? So simple. First of all, so small introduction, definition, types, parenchyma types. Now let's next one, collonchyma. Same look like parenchyma, similar, but it is the somewhat strong when compared with parenchyma. And it is also made up of layers, primary layer, secondary layer, the mainly primary layer, but the chemical made up of different. In parenchyma cells, in parenchyma cells, cell wall made up of cellulose, hemicellulose. But here, what happened is, right. Take collonchyma. This is collonchyma. But what you observe between parenchyma, collonchyma, in collonchyma, the intercellular space are very less, are absent. Intercellular space very less are absent. And if you see that this is all collonchyma. If you see the chemical structure of the cell wall, the chemical made up of pectin and cellulose. Apart from the cellulose, pectin also present. So that is the difference between collonchyma and parenchyma. And due to the deposition of pectin, it is anchored to the plant body. It gives anchoring and support to the plant body. And it mainly is not involved in photosynthesis, not involved in storage and other function. It is only the give mechanical strength to the plant body. It is only gives mechanical strength to the plant body in engage. Right? If you see the types before that occurrence, the collonchyma mainly plant body parts below the epidermis, in epidermis parenchyma. Below the epidermis present and present in stems and leaves, floral pods, fruit. So this collonchyma is generally absent in monocotyledon. That's important. Actually, if we take the plants, dicots and monocots, but generally collonchyma absent in monocotyledon. That's why the monocotyledon stems are small, mostly very weak. And types of collonchyma, angular collonchyma, lamellar coloncyma, locular. These are these three types of coloncyma. First of all, let's see angular coloncyma. Name itself given angular. What is meaning of angular? Angles. For example, this is the angles corner. So here chemical present, here chemical present. So this where the joint of the parenchyma cells, there if the chemical present, that is called angles. This is called angular coloncyma. It is mainly present in petioles of cucurbita, mainly petiole. Next, lamellar plate. This cells this type found in the tangential walls, then the radial walls. If you see diagrammatically, lamellar coloncyma, for example, this is the cell, this is the cell, cell, take color. So I already told you tangential wall, then radial walls. Here, the chemical deposited, here this radial walls, here radial walls. This is lamellar coloncyma. Next, locular coloncyma. In this type, internal spaces are seen between adjacent thickened cells. The clearly internal cells are present. For example, it look like a parenchyma cells. Here, the one cell and maintain gap either cells. It is only internal cells, internal space here. So the chemical deposition here. Intercellular space. If the intercellular space chemical deposited, this type of tissue called as locular. I told you the function colloquial tissue provides strength to the tender branches and leaves, herbaceous stem, and it gives tensile strength to the flexibility to the plant body. So this is about coloncyma. And next one, sclerenchyma type. Let's see sclerenchyma. Sclerenchyma, both for example, there is coloncyma living, parenchyma living, but the sclerenchyma tissues are not living. The sclerenchyma tissues are dead cells. And when compared with parenchyma, just let me draw here. Parenchyma cells, same. Coloncyma cells, same. Of course, this both maintains cellular, intercellular, but here the mainly difference between the chemical composition. And sclerenchyma also same, but here the sclerenchyma cells, apart from the primary cell wall and secondary cell wall also present. Primary, secondary cell walls are present. Due to the formation of secondary cell wall, it is mainly made up of pectin and lignin chemical. And due to the formation of secondary chemical, what happened due to the secondary chemicals, this cytoplasm completely utilized. Due to the utilization of this cytoplasm, completely it is dead cell. It is make a dead cell. So these tissues are called dead tissue. They are mainly found in xylem and mainly found in phloem cells. Let's see the sclerochemata cells and these sclerenchymas, one type of cells called as sclerides. But in examination, sclerides, short answer, very, very important. Sclerenchyma, if they ask sclerenchyma, you can write about scleride because in sclerenchyma, one type of cells is scleride type. Right, let's discuss scleride types. Brachysclerides, actually diagram not available, but I will draw just here. Brachysclerides, 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 macroscleride, ostnoscleride, autoscleride, and trichosclerides. We will continue later. Time is over.